Well, hi everybody, and welcome back to the show. Today I have Dr. Anitra Carr, and today we're continuing the series around vitamin C. We've had some brilliant doctors and scientists on in the last few weeks, and it's been really exciting to share some of the latest research. And we have one of our own Kiwi scientists with us today, Dr. Anitra Carr from Christchurch. Welcome to the show.
Hi, Lisa.
It's fantastic to have you. So, Dr. Anitra, can you just tell us a little bit of your background and how you got involved with vitamin C research?
Well, I first started researching back in the late 90s, so 1998, and I had just finished a PhD with the University of Otago, and I had been studying how reactive oxygen species that are produced by white blood cells react with our own tissues and damage our own tissues, because these white blood cells produce these really reactive oxidants such as hydrogen peroxide, which is hair bleach, and hypochlorous acid, which is household bleach. So very strong oxidants, and they produce these to help kill bacteria in our bodies. But these oxidants can also react with our own tissues, and that's what contributes to inflammation and the processes of inflammation. And so I've just been studying how these oxidants react with certain components in our tissues. And when I finished that, I thought, oh, it'd be really interesting to investigate how antioxidants such as vitamin C, which is one of the most potent antioxidants in our body, can help potentially protect against this damage. So scavenge those oxidants before they react with our tissues and help decrease the inflammation associated with infectious conditions. And so I applied to various people in the United States. I wanted to go to the continue my research in the United States, and so I applied to several people over there who were doing research in the area that I was interested in, and they all wrote back and said yes, we have postdoctoral positions available. And so I selected one on the advice of my PhD supervisor, and this was Professor Boltz-Frye. He was at the time in Boston, and after I said yes, I'd like to work with him, he wrote back and said, oh, by the way, I'm moving to the West Coast, to Oregon, and I'm going to be the new director of the Linus Institute.
Oh, wow.
It's very fortuitous. And I like the West Coast of the United States. I've done a bit of work in California during my PhD. And so I was quite happy with that. And so the Linus Pauling had died just a few years prior to that. And so the Linus Pauling Institute, which was in California at the time, kind of needed a new home. I think they're in Palo Alto. And so they ended up going to Oregon State University because that was, for a couple of reasons, that was Linus Pauling's alma mater. So where he had done his undergraduate research when I was still in agricultural college. And also because the library there was going to be able to host his papers. And so he has, this collection of his writings and papers is thousands and thousands of documents, because as you've stated before, he's one of the only people to have been awarded 2 unshared Nobel Prizes. So one around his work. So one was in chemistry around his work on the nature of the chemical bond, and the other one was a Peace Prize for his anti-nuclear campaigning. And so the Oregon State University Library has his complete collection. It's called the Linus Pauling Special Collection. And so I spent a few years at Oregon State University researching how vitamin C can protect against oxidation of low-density lipoprotein particles, which are what the body uses to transport fat and cholesterol around the body in the bloodstream, because our cells need cholesterol. But most people know low-density lipoprotein as bad cholesterol. I mean, it's not intrinsically bad, but if it becomes oxidized, it then can contribute to the development of atherosclerotic plaques and contribute to cardiovascular disease. And so I was looking at how vitamin C can protect against oxidation of this particle and thereby potentially protect, against development of atherosclerosis.
And what was the outcome of that? Because that would be really interesting.
So I was particularly interested in the oxidants produced by white blood cells, because these can react with these low-density lipoprotein particles and oxidize them. And vitamin C is a great scavenger, it protected it, and I was interested in how much do you need and how's it protect, is at the real biochemical level.
What's the action?
but also during this time, so late 1990s, we were interested, Professor Boltz-Frye was interested in the recommended dietary intakes for vitamin C. Because in a lot of countries are very low. I mean, these recommendations are primarily to prevent deficiency diseases such as scurvy, whereas we believe, the recommendations should be higher to help reduce the risk of chronic diseases such as cardiovascular disease and cancer and that sort of thing. That's, a better health outcome. And so in the late 1998, the Food and Nutrition Board of the Institutes of Medicine was re-examining the recommended dietary intake for the antioxidant vitamins, so A, C, and E in the United States. And we wrote a comprehensive review around, all the scientific evidence at the time for what sort of doses of vitamin C appear to protect against cardiovascular disease and cancer. And so we made a recommendation of 120 milligrams a day, which was at that time twice what the recommended dietary intake in the States was. It was 60 milligrams a day at the time. And so we submitted that document and it was considered by the Food and Nutrition Board, and also another review I'd written around vitamin C's antioxidant roles in the body versus its pro-oxidant roles. Sometimes vitamin C referred to as having pro-oxidant properties.
I've heard that,
and
to get your head around the antioxidant, it's a pro-oxidant.
It's not it's vitamin C is an antioxidant. It's not an oxidant pro-oxidant, but what it does is it can reduce, so antioxidants donate electrons and they reduce oxidized compounds. So it reduces transition metal ions such as copper and iron. So these are metals in our body that can redox cycle, so they can produce oxidants.
And we've talked about redox before on the podcast.
So what vitamin C does is it converts these metal ions into a reduced state, and then those metal ions can go on and generate oxidants.
So does it give iron and copper a longer life, does it? Is that how it gives them a iron and copper a way to keep going?
So it regenerates them so that these metal ions can keep producing oxidants. But in our body, these metal ions are all sequestered away and protected by proteins. They're not floating around free. So in the body, vitamin C doesn't seem to do that based on the evidence. It seems to just have its, true antioxidant roles, not this kind of pro-oxidant byproduct, as you might call it. So this sort of evidence was considered by the Food and Nutrition Board, and they decided, it does appear to have antioxidant roles in the body. And they also referred to Mark Levine's seminal pharmacokinetic work to kind of work out a daily dose of vitamin C they thought would be good to help foster this antioxidant potential in the body and potentially protect against these other, chronic long-term diseases such as cardiovascular disease and cancer. And so they did end up increasing the RDA for vitamin C in the States from 60 to 90 milligrams a day for men and 75 milligrams a day for women. So that was good. Not quite as high as we would have liked to see, but still a step in the right direction.
They're very conservative, aren't they? And they're slow to respond to the risk conservative. Because you'd think being in the preventative space would be a good thing if we're trying to...
Prevention is a lot cheaper, a lot easier than trying to treat a disease.
But New Zealand's even worse, isn't it? I think we're at 45 milligrams, which is
one of the lowest RDAs in the world.
That's got to change, surely.
So we're trying to generate the evidence to help, support an increase in the RDA.
Gosh, it's all so slow. Like you've been doing this for 20 odd years, and it's still.
They do say that translation of science into, medical research into clinical practice takes 15 to 20 years.
Wow, that is a really interesting statement, because this is why I think, like sharing this sort of information direct from the experts, if you like. And I said this with Professor Margaret Vissers too, like we have to make sort of educated decisions as people in trouble now, whether you've got cancer or whether, like in my case, have a dad who had sepsis, you have to make an educated decision now based on you're running out of time. And we're waiting for the research, and the research will be great, but it will be another 10 to 20 years down the line before it actually, and then, in the medical world, it seems to be a very slow, Dr. Fowler said it beautifully when I had him on last week. It's like trying to shift a super tanker, critical care he was referring to. And he says it's just very, very slowly coming around. And I know I had Dr. Ron Hunninghake on as well from the Reardon Institute, another fantastic doctor. And he talked about medical mavericks. Dr. Hugh Reardon had written 3 books on people who were really ahead of their time, got in trouble for it, and then actually the research and everything caught up with them later, So that's an interesting. So if you're listening to this, New Zealand has got 45 milligrams as the RDA, but that's just to keep you out of scurvy, pretty much. You want more. Oh, right. So, okay. So you've done all this antioxidant research and this, with the ROSs and at the Linus Pauling Institute. When did you start to develop an interest in the infectious diseases, sepsis side of that? Because I'd really love to.
Yeah, that's more recent. So, after about 3 years at the Institute, I decided to have our first child and moved back to New Zealand. And I made the decision to quit science and just focus on bringing up our family.
Yep.
I ended up having 3 children and stayed home for 9 years looking after the children. And I made the decision that they were more important than my career.
Wonderful. That's an interesting fact as well. Like as a mum and a scientist, like incredibly dedicated career that you've spent years getting to, and then trying to juggle mum roles with scientist roles and taking 9 years out of your career. Has that hurt your career massively, or been able to catch up, so to speak?
It hasn't hurt my career. I mean, I'm 10 years behind my contemporaries, my colleagues, because I took that time out. But that's the decision I made, and I stand by it because the research shows that the first 3 years of a child's life are very important. So I thought, well, I'll dedicate myself to the children in the early years.
Yeah.
And after those 9 years, I thought, right, I've done my time.
Kids.
I'm really keen to get back to
work. Mum's going to be a working mum from now on.
But I just went back to work part-time, so within school hours, so that I was still there for them after school hours. And one of the things that drew me back to work, I was recruited back to run a human intervention study. What really excited me, because when I was in the lab doing lab-based research, I always felt too removed from the people who were meant to be helping. And so I'm much more interested in the whole person and how they're feeling, not what's happening inside a single cell, which is kind of where
that makes sense.
I was really excited and really grateful to be recruited back, especially after taking 9 years out, from my career. The discoveries have been made during that time that I had no idea until I went back and I thought, okay, I've got a bit of catching up to do. And
so what was that first intervention study, that human
this was a kiwi fruit study. So kiwi fruit are very high in vitamin C, and so we were interested in knowing how many kiwi fruit do you need to eat to get adequate and optimal vitamin C levels. So it was just kind of a dosing study.
Brilliant.
Then we went on to compare kiwi fruit with tablets. So animal research had shown that food sources of vitamin C seem to be a bit better than tablet sources, and so we thought we'd translate that into a human study, and what we found is there's no difference.
No difference
in uptake of vitamin C from food versus tablets. The body is really good at because we need vitamin C. Our body has adapted ways to
take it wherever it gets it,
take it up regardless of the source.
Wow,
that's the structure of vitamin C is the same in foods as it is in tablets. So the body recognizes it the same, takes up the same amounts. I mean, the benefit of food is that you're also getting all the other vitamins and minerals and fiber. So we still recommend food, but it is, in our daily diets these days, it's very hard to get, your 200 milligrams a day of vitamin C.
Just from fruit and veggies,
just with fruit and vegetables. And as you know, different fruits and vegetables have quite different amounts of vitamin C, which a lot of people aren't aware of. People know that kiwi fruit and citrus are high, but they may not realize that apples and bananas are actually quite low in vitamin C.
Or capsicums are quite high.
Broccoli.
You wouldn't think broccoli, you wouldn't. And if you decide to take a supplement, is there a better supplement? I have heard concerns about corn-derived vitamin C because of the glyphosate discussion, and that's a bit hard to track, really, the types of vitamin C. But is there any sort of research around? I mean, I've talked previously with a couple of the doctors and scientists around liposomal delivery. Have you seen anything in that department, or any supplementation method that's better?
Not convincingly better. I mean, there might be trials that show it does slightly better than just your normal chewable vitamin C, but I just go for the standard cheap vitamin C.
You don't have to. It doesn't have to be super special. Like, it's a pretty simple molecule, isn't it? Like, the body is pretty, like you say, it needs it, it knows it.
Liposomal vitamin C is kind of wrapped up in lipids. And the body doesn't need that, because, like you say, it is designed to recognize vitamin C in its natural form in foods and such like.
So, who was it? I think Dr. Thomas Levy was saying it bypasses some of the digestive issues, because with vitamin C you can get digestive stress when you take bigger.
If you take really high doses, some people, like we're talking like more than 4 grams a day, some people can get gastrointestinal.
Get a bit of
maybe it does protect against that.
Okay, so then you've moved into, and forgive me if I'm jumping ahead, but very keen to talk about the role of sepsis and pneumonia and patients in ICU for reasons.
So after about 5 years, of doing that research part-time, I managed to get a Health Research Council, Sir Charles Hooker's Health Research Fellowship, which allowed me to move into the more clinical arena of, studying infection, which was an area I was interested in. And so we've done some observational studies where we have recruited patients who have pneumonia, measured their vitamin C levels and their levels of oxidative stress, and found that they have very low levels of vitamin C and high levels of oxidative stress. And the more severe the condition, the worse this is, the lower the vitamin C levels and the higher the oxidative stress. So if those patients with pneumonia go on to develop sepsis, and sepsis is kind of this uncontrolled inflammatory response to an severe infection, and that can develop into multi-organ failure, and the patients need to be admitted to the intensive care unit, and that can go on further to develop into septic shock due to failure of the cardiovascular system, and up to half those patients die. It's the major cause of death in intensive and critically ill patients.
And that's what I unfortunately experienced with my dad. And so with the organs starting to break down, so when you get anything like pneumonia or sepsis, the body's requirement just goes up.
It does,
100-fold or more.
At least 30-fold, yes. And so it's very hard to get those amounts into a patient orally. And so when patients are in the intensive care unit, they're generally kept sedated because they're being mechanically ventilated. And so they're given nutrition in 2 different ways because they can't eat. And so the main way is to drip feed it directly into the stomach, liquid nutrition into the stomach through a nasogastric tube. And the other way is to inject it directly into the bloodstream. And so the recommended amounts of vitamin C by these means is about 100 milligrams a day.
That's nothing.
So what we did in one of our studies was we looked at how much vitamin C these patients should theoretically have in their blood based on how much vitamin C they're consuming. Because 100 milligrams a day in a healthy person is more than adequate to, well, provides adequate plasma, what we would consider adequate plasma levels. And so we mapped out what it would look like in these patients based on how much they were getting. And then we compared it with what we actually measured in their blood.
Yep.
And it was way lower than what it theoretically should have been. And so this was an indication that, yes, they need a lot more vitamin C than they're getting in their, standard
nutrition,
liquid nutrition
yeah.
And that the body obviously has these much higher requirements, which has been shown previously by other researchers.
And so that's leading to almost a scurvy-like situation. I mean, some of these severe sepsis people, I remember seeing one of your graphs there with, the sort of normal community cohort of people, young people, middle-aged people, and then down into the more severe pneumonia and then sepsis and severe sepsis, and they are just over the scurvy level. So basically their bodies are falling apart because of that, as well as the sepsis, if you like.
And that's even on top of, being given it daily, 100 milligrams a day, or at least 100 milligrams a day. They're still really
it's just not touching the sides.
Right. And
so why is this not even for people going into the hospital? Why is it that even though, okay, we may not yet know the dosages, why is not every hospital testing, at least their really sick patients, what their vitamin C levels are, and then treating it the nutrient deficiency only, even apart from the high dose intravenous stuff, but just actually with my dad, I was unable to get a vitamin C test done to prove my case. I couldn't prove my case because I couldn't get it tested.
Yes. No, it's so true. It's because doctors don't learn about nutrients in medical school
at all.
Training. So they're not familiar with how important they are for the body. They're not familiar with all the recent research around all the different functions and mechanisms of action that vitamin C carries out. There's, over the last 10 years, all these brand new mechanisms and functions have been discovered. They think we know everything there is to know about it. But
we don't.
We're discovering new things all the time, which makes it very exciting.
It is.
And so they don't, first of all, they don't understand the hospital system isn't set up to routinely measure it. It is only ever measured if someone comes in with suspected scurvy, and even then a lot of doctors aren't used to recognizing the symptoms of scurvy. It's not something they're familiar with because, it doesn't.
They think it no longer exists because that's what sailors had in the 1800s.
It still is apparent in the Western world.
and certainly in the sick population.
Yes. But I think... So when I first applied for funding to carry out these studies in pneumonia and sepsis, there were only a couple of papers had been published at that time looking at vitamin C sepsis, and that was Barry Fowler's safety, dosing study.
That phase 1 trial.
Study, and another one, small one in Iran. And so there was very, very little information out there at the time. And so I put in an application for us to carry out an intervention study in our ICU at Christchurch. So just a small one, 40 people, 20 getting a placebo control with no vitamin C and 20 getting intravenous vitamin C. And not long after that, Paul Marrick's study came out.
Yep. I remember
that stimulated a real explosion in research in this field because of the media interest. So the media picked up on it, and it hit the world. I'd been talking about this for years to doctors, ICU doctors and that.
And not getting anywhere.
Trying to get them, news about it, but it wasn't until it hit the media and they heard about it through the media, they thought, oh, okay, maybe there's something here. So that just goes to show how important media can be for...
Exactly, while we're doing this show. Okay, I'm not CNN, but you know what I mean. We've got to get this from the ground up moving.
Yeah. And so since then, there's been many studies carried out around the world, all of different quality. So we're learning more and more information all the time. Clinical trials, they take a long time to run. Patient recruitment being the most difficult part. The other thing is that a lot of the clinical trialists, the clinical researchers, are used to running drug trials, and so they treat vitamin C like a drug, but it's not a drug. It's a nutrient, it's a vitamin that the body is specially designed to take up and use, which is very risk drugs. And so they don't always understand how vitamin C works in the body.
Yep.
And it's important to know how it's working in order to design good studies, good quality studies. So a lot of the data that's come out may be impacted by how well the study was done and thought out. So that, there's still, we still don't know, all the important aspects about, the dose, how often should you give it, when should you give it? I mean, ideally it should be given, as early as possible.
Early as possible.
As soon as they hit the hospital.
Yep.
Don't wait until they're at death's door and septic shock. It's hard for a vitamin to do something at that stage, even really high,
Amen.
even really high dose vitamin,
Yeah.
The earlier you give it, the longer you give it for. Don't just, most of these trials have given it for 4 days and then they stop.
Yeah, I've wondered that.
Continue the whole time they're in the ICU, because one pharmacokinetics study showed that when you stop that vitamin C, some of those patients just drop straight back down to where they were.
Exactly.
They need that continued input.
So why has it been made that it's only, all the studies I've seen, I think, have been 4-day, 96-hour studies. Occasionally one of them is, or for the length of stay in ICU, but most of them have been 96 hours, and most of them have been very, very conservative dosing. From what I understand, conservative dosing. And I know Dr. Barry Fowler said, there's some, consideration about oxalate and kidney function. And I'm like, but this is still that's a very low risk for someone who's got sepsis. If
a patient has kidney dysfunction in an ICU, they put on hemodialysis anyway, which clears the excess vitamin C. So it's not such an issue for those patients. But a lot of these studies were designed to reproduce the first studies that came out, to see if they were reproducible. So that's why they're using similar regimes. But now that we know more about it, each study adds another piece to the puzzle. And so hopefully future studies will, look more into what dose do you actually need.
Yeah.
And it probably varies depending on, the type of illness,
severity,
etc. How long, and I believe, once they leave the ICU, so patients who've had sepsis, who survive sepsis, they can go on to have real disabilities, problems, physical disabilities, cognitive issues, psychological issues like depression and anxiety. And so, I really believe if they keep taking vitamin C when they leave the hospital, just orally, that might help with those conditions. That hasn't been researched yet.
Yep.
So that's a whole area of research that should be carried out too.
So if I was to ask you, in your dream world where your resources were unlimited and you had lots of money and you had lots of people to help you do all these and you had enough patients to enroll, what are some of the things that you would, like as a scientist, and you understand some of the mechanisms and the cofactors, which I want to get onto as well, what are some of the studies that you would like to see happen? So we can move this along faster. What are some of the key things? So quality of life afterwards. Studies, bigger dosages. What
really practical things that the doctors need to know, I think, is what's important, like how much to give, how often to give it. Most of the studies have done the 4 times a day, because that's what was done in the initial studies. Is it better to give it continuously? So when they're in the ICU, you can just infuse drugs continuously, rather than this kind of bolus dosing. So do more research around that. So the frequency, dosing, and timing, like when do you administer it, how long should you administer it for? I mean, there's so many important aspects around that. I mean, we've got the foundational research done now. We can start teasing out the finer details now, I think, rather than just doing the same study designs over and over again.
Yeah, and reproducing it. Yeah.
Modify those study designs to start addressing these, other issues. And there's some really big studies underway at the moment, like one in Canada with 800 people. I mean, they'll give us really good information, those sorts of studies, rather than that, the little studies that some of us who live in small countries that
can't afford those
that's right.
studies that cost millions and millions of dollars. And is there a trouble with funding because of, it's not a drug that we're developing here? Is that make it harder to get funding?
It's extremely hard to get funding because, often on the assessing committee, it's, often medical people on the assessing committees who don't believe in vitamin C, have only heard the bad press or the misinformation, don't understand the importance of the relevance. And so that's where this outreach is really important. It's just educating people about the science behind it. It's not hocus pocus.
Yeah.
It's real.
Yeah, I mean, if I can share, and I've shared a little bit on the past episodes, but with my case with my dad, I know and I felt they just put me in that quackery pot, and they paid lip service to listening to me. They didn't really, but I'm quite in this case I had to be quite forceful because my dad was dying, and I didn't go away. I wouldn't go away. Most people would go away because of, I just wish I knew then what I know now, even because I wasn't that deep into the research, and now I am deep into the research and really an advocate for this. But, I was treated like, there was one really good doctor who listened to me, who advocated. He didn't believe in it. He didn't understand the mechanisms of action or any of that sciencey stuff, but he did advocate for me at the ethics committee, whereas everyone else would just roll their eyes, basically. And this is why I think it's so important to share to come back again and again to the science, and for them to just open up their eyes, just because they didn't learn it in medical school and it's not in their current textbook, for, like you say, because it takes 20 years probably to get into that textbook, that they and because it's a vitamin, they just immediately shut down, is how I felt. They just immediately went, just eat an orange, and you're good to go. I mean, the surgeon, I had a friend that was going into surgery, and she was like, should I have intravenous vitamin C before I go into surgery to prepare my body? Very logical thing to do in my eyes. Yeah. No, you don't need that. Just eat an orange, and it's like, oh, you don't get the whole why and how and what happens when the body goes through a trauma and a surgery or a sepsis or any of these things. And I don't know, like there's some bigger, issues at play with the whole pharmacological model that our whole system is built upon, and that nutrients and nutrition isn't taught in medical school. So we're up against this big sort of brick wall. And when I tell my story to people, just to, sharing with friends and things, they were going, but where's the downside? He was dying anyway. Why couldn't he have it? And I said, well, you're up against machinery. You're up against ethics committees, legal battles, and a system that is just very staid and conservative in its approach. And that's not to criticize individual people within the system. I'm not wanting to do that. I'm just trying to go, make people aware, because people go into a hospital setting or something and they expect to have the latest and greatest information available, that the doctors know all that. And unfortunately, that's not always the case. Do you find it frustrating?
Yes. I mean, it's not the doctor's fault as such, because they're very busy people. They don't have time to keep up with all the literature, and they're not likely to be going into the nutrition literature in the first place, which is why we try and publish as much of our stuff in, the clinical literature. They're more likely to see it then. And so they have their patients' best interests at heart. They've just heard the bad things about vitamin C and the misinformation. And so they don't want to do harm to the patient, I guess, is where they're coming from. And they don't have time to read all the latest information. And that's why just piece by piece, kind of chip away at that and, you know, educate them. And, you know, hopefully it'll come into, you know, the training of, you know, the new doctors in future. Hopefully more nutrition courses will be introduced into medical training, because it's not just vitamin C. You know, the body needs all the vitamins. They're all vital to life. That's where the name comes from. You don't have them, you die. It's as simple as that. So yeah, I think it's vital that this information gets into, you know, into the appropriate arenas.
Yeah, and I think that's why, you know, I'm passionate about the show, is that, you know, my sort of outlook on the whole thing is, yes, I'm not a doctor, but I can give voice to doctors and researchers, and I can curate and I can investigate and I can share. And, you know, this is a very emotional topic for me, or for obvious reasons, but I'm trying to take the emotion out of it, because that doesn't help the discussion. And it's really, it's hard, but I understand the importance, because you don't want to, if I know that if I share things in an emotional manner, then I'll get shut down as having mental health problems and being a grieving daughter, when actually, no, I'm an intelligent person who's educated herself in this, and I've got the best people and the best researchers and the best scientists and the best doctors sharing their latest research. And I hope that by doing that, you can get, you know, one mind after the other, and just get them to understand, rather than the emotional side of things. Because what I do want to also share with the story is that every person's life that is saved is a family that's not grieving. You know, these are not statistics. When Dr. Barry Fowler's research or Dr. Merrick's research, and you see a drop from, I think Dr. Merrick's was 40% mortality to 8%, and Dr. Barry's was something like 49 down to 29. Don't quote me on the numbers, but big numbers in drops in mortality. And you go, those are just dozens, or not, if not hundreds of lives that are, you know, are saved, and those families are saved from that grief, you know. And worldwide there is something, well, I've heard a couple of estimates, between 30 and 50 million people a year who get sepsis.
Right. Yeah.
Of those, you know, 1 in 5, I've heard in your research, 1 in 5 in New Zealand ICUs dies of sepsis. This is a huge problem. This is as big as cancer, and actually is one of the complications often of cancer therapies.
Yeah.
So I don't think people understand the enormity of sepsis itself, and then, you know, pneumonia, and then we can go into the discussion around COVID and cancer and all of those other things. It's like we're talking millions of lives every year around the world. So this research is just absolutely crucial, you know. And sorry, I've gotten on my bandwagon a little bit, but I really want to get this information out there, and that I think it's really, really important. Let's change tack a little bit and just talk a little bit briefly, because I haven't covered this subject with the other vitamin C interviews that I've done, around the cofactor. So vitamin C is a cofactor for so many different areas. So I remember from one of your lectures, it has epigenetic influences. It has, like, with collagen synthesis, and that's not just for your skin and your hair and your nails, but also has implications for cancer. You've got your HIF, which Professor Margaret Visser's talked about, your hypoxic inducible factor, tumor growth. Can you just go and give me a little bit of information around the vasopressor one? It would be very good. And anything else that pops to mind there.
Yeah, so a cofactor is a compound that helps enzyme function. So everything in our cells relies on the functions of enzymes to carry out reactions in our cells. All the chemical reactions require enzymes. And so a cofactor supports that function. And so early on, when I was just starting in this area of research in the field of sepsis, I was looking at the different cofactor functions of vitamin C, and one of them is a cofactor for the enzyme which synthesizes noradrenaline.
Mm-hmm.
And noradrenaline is one of the main drugs, as you might say, that's given to patients who are going into septic shock. So it's given to the patients to try and increase their blood pressure, and it works by making the muscles around the blood vessels contract.
Yep.
Makes the blood vessels a bit smaller, and so increases your blood pressure. And so vitamin C is a cofactor for the enzyme that naturally synthesizes noradrenaline in our body. And there's another enzyme which synthesizes hormones, one of which is vasopressin.
Yep.
And this is another drug that's also sometimes given to these patients to help increase blood pressure. And it works by increasing the reuptake of water by the kidneys, so that increases your blood volume and hence your blood pressure. So, for a lot of ICU patients, they're given noradrenaline, and sometimes they're given vasopressin on top of that to, really try and get their blood pressure up.
Because they're collapsing their cardiovascular system.
And I realized, oh, wait a minute, vitamin C is also a cofactor for this enzyme that synthesizes vasopressin. So here it is, a cofactor for two quite different enzymes that synthesize vasopressors naturally in our body. And so if these patients are coming into the ICU very low in vitamin C and going into shock, is one of those reasons because they don't have enough vitamin C in their body to support natural vasopressor function. Hence the doctors, have to give them these drugs. But if we're able to give them vitamin C early enough it could potentially support their own natural synthesis of these vasopressors in the body, which is a much better way to do it, because if drugs are given, from the outside, they're often given in high doses and not regulated, and so can cause side effects. Whereas if it's being produced in the body knows what it's doing. It regulates how much and how often, all those sort of intricate details, and so you don't get the nasty side effects.
Can I share a bit of a story there? Because both my mum, in her case, where she had an aneurysm 4 years ago, she was on noradrenaline, and it could only be given in the ICU. And originally she was in the neurological ward, and they couldn't do it there. And I only realized, she was going into a coma. So she had massive brain damage, going into a coma. When they took her up to ICU, they could give her the noradrenaline. That opened up the vessels in the head a little bit, or kept the pressure up so that the vessels were open, to stop the vasospasms in her case, which was killing parts of her brain. But she'd been in the neurological ward where they couldn't give any of that earlier, and so the damage had already been done partly. And then with the case with my dad, back then I didn't know anything about vitamin C, of course. With the case with my dad in July this year, he was in. I got vitamin C, but it was on day 13 of his 15-day battle, because I had to go through ethics committee and all of that sort of jazz. So he was an absolute death's doorstep, should have been dead days ago, according to the doctors. They couldn't believe he was still going, but he was one tough man. Right. So I don't know how he was still alive, but he was. And the very first infusion that we got of vitamin C, immediately we were able to take him off NORAD for a period of about 8 hours. We needed the vitamin C again. It took me another 18 hours before I could get permission to get the second one. Unfortunately, I couldn't get it in the 6-hour bolus, which would have been ideal. We gave him initially 15 grams. So this was, again, multiple organ failure, fecal matter in the peritoneum, desperate straits. His CRP, his C-reactive protein, dropped from 246 down to 115. His white blood cell count improved, and his kidney function went from 27% to 33%. And they were able to take him off vasopressors and noradrenaline for about 8 hours. That is incredible for someone who could die at any moment. And we failed because I struggled to get the second, I struggled to get the third infusion, and it really was too late. But even at that point, I thought that might be interesting for your research. I have all the medical records, by the way, if you want to have a look at the data exactly, but it really was a strong, he doesn't need the noradrenaline, his blood pressure was going up, and that's a really good sign, you know, as is the dropping of the CRP, which was still very high at 115, but it was way better than where it had been. So goodness, what would have happened if I'd had it on day one from the surgery?
Exactly.
Yeah. And none of that is understood, you know. So that's one of the cofactors and that brings to mind, just as someone who's connecting the dots, if you have an HPA axis problem, like your adrenals aren't doing their job well and your cortisol, vitamin C would probably be a good thing to take to support.
It's sometimes referred to as a stress hormone because it is involved in the adrenal response. So, and people who are under stress, you know, or in animal studies where they've stressed animals, they appear, you know, they use more vitamin C, generate more vitamin C. The animals that can synthesize it themselves, they generate more vitamin C to compensate.
Exactly. Yeah.
That.
And
we can't make our own anymore, so we have to take more if we're under stressful conditions.
Exactly. And that's a really, you know, it's just a funny thing of evolution that we've lost the ability to synthesize more as we like animals like the goat, especially, it can synthesize like a ton more when it needs it. We were given big brains so that we could make vitamin C so we could take it. What are some of the other cofactors, you know, just as we start to wrap it up, but just a couple of the other important cofactors. Collagen. Why is collagen important, apart from you want nice skin and hair and your joints? What I did hear in one of the lectures about collagen helping stop metastasis of cancers.
Right. Yeah, that's one mechanism. It's also very important in wound healing.
Mm-hmm.
And interestingly, a reasonable number of surgeons are aware of this, and they're a lot more open to people taking vitamin C around surgery, before and after surgery, just to help with that wound healing.
Oh wow.
Yeah, which is great. And
oncologists, are they sort of open to you know, less so? Yeah. In fact, I've had friends who've told if you take intravenous vitamin C, we won't do any treatments. And that
is primarily around all the misunderstanding around those early trials around intravenous vitamin C in cancer, where Linus showed an effect of intravenous vitamin C. The clinicians at the Mayo Clinic who tried to reproduce those studies, they used oral vitamins. Wider doses, so just small doses over a day. But back in those days, they weren't aware of the different pharmacokinetics of vitamin C. They thought oral and intravenous, they'll just be the same, like with the drug. But it's quite different. Oral uptake's a lot lower, much smaller amounts are taken up, versus intravenous. You can get really high doses in very quickly.
Up to 200 times. I heard Professor Garby Ducks saying that intravenous is up to 200 times for short periods, but that short periods makes a difference because you can get that into the tumor cells, into so that and this is the problem. Professor Margaret Vissers was saying too, you know, the original controversy around Linus Pauling's work, and because they didn't have an understanding of how could possibly this mechanism of action be working, they just poo-hooed it basically, and it caused this big rift, those on this side and those on that side. And for the next 40 something years later, still actually it's problematic.
Well, it wasn't really until Mark Levine did his, really detailed pharmacokinetic studies that people realized the big differences between oral and intravenous. And also, these more recent discoveries of vitamin C's cofactor functions around regulating genes through HIF and through the epigenetic enzymes. These are all mechanisms which could be involved in its anti-cancer mechanisms as such. And so, it's the epigenetic areas. It's very, very exciting, very interesting. area of research, and I think it'll enable us to personalize medicine, you know, in the future.
Yeah, I mean, I have an epigenetics program as one of my health programs, and yeah, that's looking at, okay, how are genes being influenced by your environment, and let's optimize your environment to your genes. And, you know, the vitamin C, so the vitamin C helps, so to give people an understanding. So is vitamin C helping to produce the enzymes that read the DNA and then therefore having the reactions? Is that how it works?
It works as a cofactor. So yeah, so it helps the function of the enzymes which modify the DNA. So epigenetics, so genetics is about the DNA itself. Epigenetics is above the DNA. So it's a way to regulate the DNA, as you know.
Yeah.
Usually through adding methyl groups to the DNA, adding and subtracting, and that affects how the DNA is read by the enzymes that read DNA and transcribe.
So it's turning them on or off for the simplified.
So vitamin C regulates the enzymes which modify the methyl groups and stimulates them coming off, or, you know, stimulates different mechanisms happening. So switching genes, certain genes on, switching certain genes off. You know, it can potentially regulate thousands of genes in our body through its stimulation of these enzymes.
Wow. So yeah, I've heard somewhere, I think it was 7,000 or 8,000 genes that are possibly affected by this. So we're really at the beginning of the vitamin C journey as far as the epigenetics mechanisms is concerned.
For sure.
Yeah, that's exciting.
It could explain, you know, a lot of its functions, not just in cancer, but in, you know, in all areas of health and disease. These functions could be playing a role. So yeah, huge areas of research possible there.
Yeah. Is that I remember Professor Margaret talking about TET? Is that one of the enzymes, the TET1?
Yeah, TET enzymes, that's right. Yeah.
And that's important for cancer.
Well, they're the enzymes that modify the methylated DNA, so you know, regulating the epigenetics, and yes, they're definitely involved.
They're implicated in the cancer process. Wow. Okay, we're getting quite technical here. Dr. Anitra, I just want to say thank you very much for your dedication, because I know I've listened to a couple of interviews with you, and you've actually sacrificed quite a lot to do the research that you're doing, because there isn't a heck of a lot of funding and things out there. So thank you for doing all that. It's a labor of love, I can imagine. It's a long, slow process, getting the information, getting it to be watertight, scientifically watertight, so that we can actually get people help who need help. And that, at the end of the day, is the reason I'm doing this podcast, and it's the reason you're doing your research. And hopefully, you know, together and with many others, we can you know, move the story along so that people get helped. Is there anything that we haven't covered that you think would be an important message for people listening today?
Well, I think, I mean, of course, infection is very relevant these days with COVID.
Yeah.
And so there's a lot of, you know, information and misinformation floating around out there about vitamin C and COVID. And at this stage, you know, the studies are still at the really early stages. Paul Merrick has done a wee study which shows that patients with COVID have, in the ICU, do have low vitamin C levels, like, you know, other septic patients. COVID is a severe respiratory infection, like, you know, like pneumonia. You know, pneumonia and sepsis are complications of COVID. And so I think that the key is to, you know, stay healthy, you know, eat a good healthy
your immune system,
yeah, to support your immune system. It doesn't mean you won't get COVID, but it may decrease the severity and the duration, so it doesn't go on to become the more severe version, you know, the pneumonia and sepsis. So I think that's an important message. And if you do get the infection, your requirements do go up. So you do need to take more vitamin C. You need to take gram amounts rather than milligram amounts.
Exactly.
Once again, to help, prevent it getting even more severe. So, I'm all for prevention as much as possible, not leaving it till it's too late. So I think, just look after yourselves, eat well.
Yep, and get your vitamin C. Go and buy some kiwi fruit and some oranges today, and some lemons, and capsicum, and some supplements maybe. Just as a final thing, you yourself have a study that's currently underway, which is really, really exciting. And this is based in the Christchurch Hospital, I believe, in 40 patients with sepsis. Can you just tell us a little bit the parameters of that study and when you think you'll have some results from that?
So this was patients with septic shock. So once again, at the severe end.
Yeah.
And they were administered either a placebo control, so half the patients, and the other half were given intravenous vitamin C at a dose of 100 milligrams per kilogram body tissue per day, which equates to about, 6 to 7 grams a day. And the reason for that, I had wanted to use the higher dose, very Fowler, but the ethics committee, because when I put this into the ethics committee, there were only the 2 studies out, which was Barry Fowler's and the small study out of Iran. And they said, well, slightly more people have received the lower dose versus the higher dose. So we'd rather you use the lower dose, even though there'd been no adverse events at any doses. No. And subsequently no adverse events in any studies.
No.
And so, we've used the lower dose. We've only just finished recruiting the last patients. It took a while and we had, issues with lockdown. And so now we're in the process of analysing the samples that we've collected, analysing the data. And so hopefully we'll be able to put all that together, sometime next year and publish the results next year.
Brilliant. I can't wait to see that. And that's a little bit frustrating because I would have liked to have seen a study with, the 15 to 18. And even that I thought was still very conservative, compared to some of the cancer dosages. But I understand from what Dr. Barry Fowler said, because of the decreased kidney function often in septic patients and so on. But it's just like, but they're dying, often. And it's because that was one of the arguments that was thrown at me. I could damage my dad's kidneys. The sepsis was doing that quite nicely, thanks, and he was dying anyway. So why the hell, not? But I think even at those dosages, we will hopefully see some fantastic results come out of that, and hopefully in future we'll be able to do slightly more high-powered dosages.
I think, well, the key is also the size of the study. Our study is very small. Yeah. And we were interested in being a scientist, I'm interested in how it's working in the body. Because once you understand how it's working, it's makes it easier to design better studies and future studies. And so, our study will be too small to show, yes or no, it decreases mortality or not. That we're leaving that up to the large studies to show that, and hopefully we can put a bit more science behind how it's working and what's happening in the body.
And it's such a complicated thing to design a study. I think people don't probably realise how the parameters and the limitations and the number of variables that you can look at, and the primary outcomes and the secondary outcomes and so on.
And sepsis is such a complex variable. And each person that comes in is unique in their situation. So there's huge variability in the data, and that's where the bigger studies are good, because that helps decrease, improve the statistical analyses of those studies. So I'm looking forward to the results of the big studies coming out. Yeah,
but these smaller ones are really, really important too, and it's great that we've got one going in New Zealand. So thank you very much for your work, to Dr. Anitra. It's been absolutely fascinating, and thank you for your dedication to this. I really, really appreciate you.
Thank you for inviting me.